So, you're awake.
What are we doing here?
Why are you dripping us here?
Well, you were going to stumble upon my plan.
What plan?
Well, I'm glad you asked. You see, I hate spiders. I hate spiders. 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 And now I finally have a way to get rid of them for all time.
How?
The Crispr-Cas9 endonuclease system arose from bacterial and archaeal immune systems. Bertha? When a piece of foreign DNA enters the cell, a segment of that DNA is incorporated into the bacterial genome. This is called the CRISPR locus, which contains short repeats of CRISPR DNA. The CRISPR locus is transcribed to form CRISPR RNAs. These are complementary to another segment of that same foreign DNA upon re-entry to the cell. The CRISPR RNAs act as a guide, bringing in the Cas9 endonuclease to cut the foreign DNA. This protects the cell from that DNA, which may belong to a pathogen. Humans have now adapted this mechanism for our own needs and eukaryote systems. The endonuclease enzyme Cas9 acts as a pair of molecular scissors, along with a guide RNA which is pre-designed. Together, these are called the CRISPR-Cas9 complex, which can be injected into developing embryos. Once inside the cell, the guide RNA helps Cas9 to find a DNA sequence complementary to the guide RNA. Once the sequence is found, Cas9 unwinds the DNA double helix at that point. The guide RNA binds to the target sequence. The Cas9 then creates a double-stranded break in the target DNA. Bertha? Bertha! Repair enzymes close this double-stranded break. This could occur via non-homologous end joining, which is the joining of the free ends of the DNA. Pieces of other DNA can be incorporated between the blunt ends. Or this could occur by homology-directed repair, in which the cell uses a region of homologous DNA as a template to repair the break. The repair enzymes recognize the two areas of homology in the other piece of DNA. The DNA between the areas of homology on this template strand is replicated to join the free ends of the broken strand. A gene drive allows the propagation of a particular allele through a population, even if that allele is associated with reduced evolutionary fitness. Take gene X, for example, which has the alleles XA and XB. A particular individual is homozygous for gene X possessing XAXA. A gene encoding the CRISPR-Cas9 endonuclease system can be inserted into XA, disrupting XA function. The guide RNA for the endonuclease system is complementary to a region of XA, and when expressed, a cut will be introduced there. The cut is repaired, usually by homologous recombination. As there are two regions of XA homology either side of the endonuclease gene, the endonuclease gene is copied during repair. This allows efficient knockout of XA that will be passed down to future generations. Cargo genes can also be included with the endonuclease gene, allowing the spread of certain alleles through the population. Consider a spider zygote called Bob. He is a male, therefore has one X and one Y chromosome. With targeted CRISPR-mediated changes to the Y chromosome, Bob can be changed to Jane. Jane can then breed with another male, forming offspring with the genotype XX and XY. In the case of XY offspring, the CRISPR-Cas9 endonuclease gene is encoded by the X chromosome. Therefore, these targeted changes could occur and alter the Y chromosome to X. The new genotype is XX. In a few generations, the proportion of females to males in the spider population would eventually become so high that the spiders would die out. So, do you like it?
Oh yeah, I can see how that would work.
No, there's consequences.
I thought this was Contemporary Art 101.
Well, regardless of what you think, I'm going to go ahead with my plan anyway.
Stop. Agent report from the RSPCA and you're under arrest.